So we welcome you today. Our speaker today is Dr. Larry Rizzolo. He got his PhD in biochemistry from Duke. We have to forgive him for that since I'm a Carolina person, but it's okay. He got postdoctoral training in biochemistry at Harvard and in cell biology at NYU. He joined the Yale faculty in 1993, where he served as the director of medical sciences for the section of anatomy and course director of human anatomy and development for first-year medical students. He held joint appointments in surgery and ophthalmology at Yale. He served in several research committees and served also on the editorial board of several scientific journals. He is one of the four Rizzolos who have spoken here. We've heard from Larry, we heard from Dr. Carol Rizzolo, and we've heard from two of his three sons, Greg and Doug. So we're waiting for the fifth Rizzolo to round things out. We're looking forward to that. The title of his presentation today is Tissue Replacement Therapy for Age-Related Macular Degeneration. Please welcome Dr. Larry Rizzolo.
Thank you, Bob. Is that too loud? How does that work? Okay. I don't know what this thing is doing here. There we go. Okay, good. I've been here for, I don't know, 20 years, whatever. And this is the view from my house at 5 Thompson. You're welcome to join me on the porch anytime to discuss this talk or anything, provided you can find it. I'll give you a hint. This grassy strip is Thompson Avenue. Okay. And somehow the slide doesn't want to advance. Let's try it this way. Okay, so some of this talk is derived from this review article that I wrote with some of my colleagues. It's gotten very good reviews, but the reviewers did ask for some changes. And so this talk will be posted on our YouTube channel, but not until the paper is accepted. But if you really want a recording for your private personal use, you can email me and I'd be happy to send it to you. Okay, so macular degeneration. It's a disease of aging. They're related diseases. How many people here know someone with macular degeneration? All right, just about everybody. I mean, so especially in the Chautauqua community, maybe that's why I have such a good audience today. But this photo tries to emulate what a person would see. The face is totally unrecognizable. And all of this out here is actually normal vision. Okay, so this is a disease of central vision, your ability to recognize faces or do critical things like read the Chautauqua Daily. And you could demonstrate this to yourself by covering one eye, don't squint, cover one eye, and use your thumb to block out the face, the splotch, okay? And keep focusing on your thumb, and you'll see that macular degeneration up here is very fuzzy. That's because most of the eye has to do with light, motion detection, low light signaling, seeing at night. So people with macular degeneration can walk around the room, and they won't bump into things. They can get up at night and go pee without bumping into things any more than the rest of us would do. But they can't do these critical things that what most of us would consider to be seeing. So here is a summary of clinical trials, just a handful of the ones where people are actually using stem cells. In my early talks here, I would talk about the promise of stem cells. Well, now actually stem cells are being put into use, if only in experimental ways. Now, one of my early mentors told me that, a slide like this is an affront to the audience. So I'm going to cover it up with this eye chart. And this chart was designed to help measure the visual acuity of patients with diabetic retinopathy, another major disease. And this was co-opted by the people studying macular degeneration. The idea is how many of these letters can you read? And then you have whatever medical intervention, and then ask how many letters can you read, and the difference is a measure. So if you could read 10 letters, and after the intervention, you could read 20 letters, that's an improvement of 10 letters. Yeah, that's how this data is reported. And 15 letters is considered significant. So the disease isn't that, I mean, the measure isn't that sensitive. So you need an improvement better than that. So let me just show you some of the, I'll read this because I know you can't read it. In this study, we had 9 people. Here we had 12 people. So it's small numbers. These only 2 people. Out of 6 of 9 patients showed any improvement in 11 to 15 letters. So actually, not in the realm of significance. This study had 21 and 29 letters, 2 out of 2. So that's pretty good. But it's only 2 people. 1 out of 4 improved 17 letters. 2 people, no change. Okay, so the point here is the numbers are small and the success rate is modest. Only a fraction of people show any improvement at all, and the improvement they show is borderline. So, why are we excited about this? So, we have to ask a couple of questions. What is the macula? Why are we excited about this limited success? Let me say at the outset, these are phase 1 trials, which means to measure safety, not necessarily to measure efficacy. And the good news is there are no eye tumors, no systemic tumors, no eye diseases of various sorts that might be anticipated. So the procedures from that perspective seem to be safe. But again, we have limited success in vision improvement. What are the challenges? And how do you make a replacement tissue from stem cells? So those are the things we need to explore today. So this diagram of the eye shows the pupil out here, a lens. Here's the optic nerve way back here. And most of these terms you don't need to know. If you need to know them, I will mention them many times, okay? So I'm showing a rodent eye, the lens of a rodent, because that's an experimental model, and the lens of a human. So this chamber is known as the vitreous chamber. In the human, it's big. In the rodent, it's filled with the lens. And the problem is, if you're going to do a transplant, you can't scratch the lens, okay? Otherwise, you'll get a cataract, and then you can't see what you want to do. So let's say a little bit more about this diagram. So the retina is basically a 2-layered cup. So if you follow this black line around till it turns blue, and now it folds back on itself, then it thickens. This red part is the neurosensory retina. So we have this 2-layered cup. Think of it like a balloon, and you put your fist into the balloon. And so your wrist is where the pupil is, and your fist is here in the vitreous. And so one layer of the balloon is pressed up against the other to create a potential space. Right? What's a potential space? You look at my shirt and my skin, there's no space between my shirt and skin. Actually, there's a potential space because I could slip my hand in there and create a real space that's holding my hand. Okay, I didn't have to rip any tissues. I didn't have to damage anything. Potential space, now I fill it, it's a real space. Okay, so, and we're going to take advantage of that anatomical feature. Now, we talk about the retina. It's composed of both layers of the cup. The outer layer that's black is the retinal pigment epithelium. It's called pigment epithelium because it's filled with the same pigment found in your skin. There's just such a concentration of it that the pigment epithelium is black. The red part is the neurosensory retina. And the space between them is the subretinal space. Subretinal space, subretinal space, subretinal space. Okay, something you need to know. Because what we're going to do is we're going to insert a needle, a cannula, into that potential space and make it a real space by injecting fluid. Now that we have a real space, we can insert our tissue into it, either as a cell suspension or as a solid tissue. Um, and then the pigment epithelia, what its big job is to pump water out of that space. So the retina always stays detached. So we create what we call a serous meaning water, serous retinal detachment. It's a temporary thing because the RPE will pump that water out, and now your implant will be sandwiched between what's left of the neurosensory retina and the choroid, because the reason you did this procedure was because the RPE was degenerated. Okay. Back here is the macula. So we said, what is the macula? If you look at the visual axis, light comes in through the lens, straight through the lens, and hits this spot. Macula means spot. And it's the retina thins a little bit at that spot. And so in a human procedure, we'd want to put the needle through this way, through the vitreous. Actually, we take the vitreous out and replace it with saline. Inject fluid, raise the macula up to create the serous retinal detachment, and put our tissue in there. So that's the thing. So I need to explain a little bit more about are the retina and choroid. So I'm going to look at this box, this dotted box, and I'm going to expand those in the next slide. So here's the retina. The retina is normally clear. It's translucent. So light's coming in through the top of this image, past all these layers down to the pigment epithelium. And so to be able to see, we had to stain the tissue. So we could find this laminar structure, 3 rows, 3 lamina layers of nuclei, and they're all sending projections into these intervening spaces where they synapse. Okay, so a lot of terms here. The only things you need to know is one very fundamental thing is that photoreceptors, we have 4 types of those. There are 2 types of horizontal cells, 15 types of bipolar cells, 70 types of amacrine cells, and 20 to 40 types of retinal ganglion cells. What is going on? So light comes in. If it's not captured here in this outer segment, it'll be absorbed by the RPE, so we don't have backscatter. So light gets absorbed here, sets an electrical signal that's transmitted to a bipolar cell, that's transmitted to a retinal ganglion cell, and then to other parts of the brain. But before you get there, you have these horizontal cells that are going to spread the signal laterally. And then when we get up here, we're going to have these amacrine cells that are going to spread the signal laterally. All right, what's this all about? We have a complex circuit here that we barely understand. It's like a computer chip. Think of your phone and think of your camera. Okay, the old days, you took a camera, you had a piece of film. The retina is not at all like a piece of film, by the way. So you have a piece of film, and if you shake, the image will be fuzzy. If it's out of focus, the image will be fuzzy. But in your phone, you don't have a piece of film, you have a computer chip. So I can even pan and do one of these panorama shots, and even though my hand shakes a little bit, the computer chip figures it out. It gives you a nice, flat, in-focus image. So that's what this retina is. It's a computer chip. It's processing the signal. Signal processing begins here. This is part of the brain. Okay, it's the only part of the brain that you can see without cutting anything open. Just look straight in the eye, and this is what you see. And I'm not going to go into the optical techniques that have been devised that you can see evidence of these layers and study what's going on with your patient. Okay, let's expand this area down here. And this is what we call the outer blood retinal barrier. There are 2 blood retinal barriers. One is blood vessels that are up in here. They're taking care of the health of the cells in these layers, but they don't take care of the photoreceptor layers. Rather, it's vessels in the choroid, we call the choriocapillaris, and they're unique in the sense, or specialized in the sense that they have these windows called fenestra. Fenestra means window. Okay, so they're very leaky. And so the barrier, the blood-brain barrier, is actually formed by the pigment epithelium. Here we have inner segments of the photoreceptor. Here we have outer segments. Remember, this is where I said the light is captured. And every day, these disc membranes are shed from the tip, and new disc membranes are added at the top. So think of a stack of coins. Every day, you add some coins to the bottom of the stack and remove an equal number of coins from the top of the stack. So the number of coins stays the same, but they're constantly being renewed, because light is oxidizing and it's damaging, and you need a way to routinely replace these. So what happens to the shed discs? The pigment epithelium eats them and degrades them. And it does this for your entire life, because these cells last your lifetime. They're not replaced like blood cells are replaced every 120 days, and the skin that you're looking at me is replaced every 30 days. These cells have to last your entire life. And so after time, this process deteriorates, and that leads to disease. So we have this feature here, the Brooks membrane, made up of many layers. You don't need to know the layers. What you need to know is I'm going to describe scaffolds, artificial scaffolds that are designed to replace this rather complex structure, whose function is we're not entirely sure. Okay. So what happens in macular degeneration is this pigment epithelium gets sick and dies, and the surrounding tissues get sick and die, because these interactions, that's what these arrows are supposed to be telling people about. This is a functional unit, and if you make the choriocapillaris sick and it dies, the other two tissues get sick and die. Any one of these tissues, you make them sick, the other tissues die. Okay, so in trying to figure out a disease, you have to figure out what is the primary event and what are secondary events. So in most forms of macular degeneration, it's either the RPE or the choroid is the primary event. Okay, sometimes my advanced doesn't want to work. So here's a nice picture. This is a picture of chick RPE. And the neural retina has been actually removed. So I could see these microvilli. Oh, that's an important point. So these microvilli surround these outer segments. So the subretinal space, the potential space, lies between the microvilli and these outer segments. Okay. So I've used a stain that stains those red. I've stained the lateral membranes, basal membranes, with a stain that is green. The finding here was the green was not expected to be at in this location, so that was an interesting finding, a story for another day. This is a choriocapillaris. This is a blood cell in the choriocapillaris. This thin space here is Brooks membrane. Okay, you can't see it because it's not labeled with anything that you can see. Okay, so here's what happens in macular degeneration. You gradually lose the pigment epithelium, where you had many layers of photoreceptor cells. You end up with a single monolayer, and eventually, single monolayer of cells, and eventually they disappear altogether. The rest of the retina stays intact, at least for a little while. And actually, it can stay intact for extended period of time. But all this circuitry, all those neurons I told you about, disorganize. Apparent synapses, totally disorganized thing. And there's an open question. If I replace the photoreceptors, would those neural circuits restore in a functional way? What people do nowadays for clinical trials is they treat patients out here like this. I'm going to focus today on transplanting pigment epithelium. And out here, pigment epithelium will have no beneficial effect. There's nothing there for the pigment epithelium to interact with. So you need a retina that's not so far gone. It still has a little bit left. And that's unethical. Okay, so if you join a clinical trial and you have one eye that sees okay, it's disease, but you still have some vision, and the other one is completely shot, they're going to use the other eye for the trial, right? Because we need to know more about what's going on before we can risk blinding somebody with a risky procedure. So the implant is put more or less here, and most of the implant is not where it can do any good, but some of the implant is out here where there might be a few photoreceptors left. So that's why visual improvement in those clinical studies that I just told you about are so poor. So when can we get to the point where we feel comfortable using an eye that still has some functionality left to it? Another area of research that I'm not going to go into very much today, I can mention a few things in questions if you ask later, is what do we do about this eye where things are far gone? Okay, and there's some approaches to what we might do in that circumstance. Okay. Okay, so stem cell derived retina. How do we do it? So we have stem cells. What are stem cells? They are a group of cells that can divide and reproduce themselves forever and have the potential to give rise to many different tissues. Okay. In the adult, we have stem cell populations in our gut, for example. We have 4 types of cells lining our gut, and there's a stem cell that can make those 4 cells. So they will divide and divide, give off some daughter cells to
Zoom
stay stem cells,
ratio.
and
That's
other.
not it. So how do I get sound?
That's what I'm having a hard
Start
time.
video. I'm on
Okay,
mute.
so someone might be not on the Zoom, might not be muted. Teresa, if you could find those people and mute them. Okay, so so the stem cell population never exhausts itself. It's always self-renewing itself with some daughter cells, and some daughter cells are differentiating into some number of neurons. So we take our stem cells. These are pluripotent stem cells. That means they can give rise to just about everything, except the placenta. Okay. Of course, in tissue, we can push things around and make them generate placenta too. But anyway, what we're going to do is put them in a media that's going to push them to form anterior forebrain. So we've come up with this cocktail of goodies that will accomplish that. And then we see something remarkable. We form these little spheres, shown in cross section here.
This is
and
unmute.
accumulating on one part of the sphere are these green cells. And these green cells are self-selecting. Okay? They're the cells that are going to become the eye. And magically, they will form a little bleb, a little outpocketing, forming that balloon I was talking about. And push your fist into this balloon, and you get this 2-layered cup. So this happens, so you give the right growth media, you grow the cells in cocktail A for 4 or 5 days, and then switch to cocktail B with different set of growth factors and so forth, transcription factors, and then wait a little bit, and then change media to cocktail 3, and you could push down this way and make these things. And then what you do is cut this off right here, and the RPE will snap back and form a little plug. So think of a vase with, or an urn with a cork in the top. So the cork is the RPE, and the urn is the retina, will become the retina, and that looks like this. Okay. So these were published by a buddy of mine. You can see the pigment epithelium. It's pretty black, right? And it's surrounded by what will become the neural retina. So here is an enlargement. You can cut here and harvest the pigment epithelium and culture it by itself, and the rest you can culture the presumptive neural retina. And this outer layer will be the photoreceptor cells. This inner layer will be those retina ganglion cells, and we have all the layers in between. And this remark works astonishingly well, at least to a point. So let's look at these retinal spheres. And this is an early stage in the process. We've labeled the nuclei so you can see every cell. And then these images are just taken with different filters. So we've put in a stain here that reveals one type of cell, another stain that reveals a different type of cell, and a third type of cell. And when you merge all these together, you can find these layers that resemble to some extent the retinal layers. If we look later in the process, so now these have been in culture for maybe 6 months, let's say. We have these photoreceptors. You can see these little projections out. They're making inner segments and little bits of outer segments. You're seeing the cell bodies here where the nuclei reside. And we're seeing here in this panels 2 types of opsin. S-opsin is short wavelength, so that's detecting your blues, and your middle and long range opsin. This antibody doesn't distinguish between those 2, but they see the rest of the color range. Okay, so so these are pretty sophisticated tissues. Unfortunately, the inner layers, the ganglion cell layer here, is degenerate. Okay, and we don't know why that happens. We have suggestions for why that happens, but this isn't the best thing to use for a tissue replacement tissue. It's good for studying what's going on, testing drug therapies and stuff like that. And I'll show you why in a moment. So here is my RPE from human-induced pluripotent stem cells, okay, cultured on a polyester membrane. So the polyester is taking the place of that Brooks membrane. Okay. So now we're looking down on that membrane, that scaffold. So think of it as a flat sheet. We're looking head on the flat sheet, and we've stained in blue the nuclei of these cells, and in red 2 different kinds of protein that should be found not only on the lateral borders, but on what we call the apical side of the lateral borders. So what do I mean by that? Let's take this flat sheet and turn it 90 degrees. So now we're looking on the edge, and we can see the cell nuclei. The filter is down here. You can't see it black because it's not stained. And we have these 2 proteins found between the nuclei and on the apical side. The apical side is where the microvilli are that would interact with the photoreceptors. And this is just an enlargement showing that. So the problem is, if we take one of those retinal spheroids and we layer it on our culture, notice only a small fraction of the surface of the retinal organoid is actually in contact with the RPE. And this would be the same problem if you injected this thing into the subretinal space. The ganglion cells are out in here. The photoreceptors are out here. We can make an inverted spheroid, but then you have a problem that the photoreceptor cells can't interact with the RPE. So it would be desirable to make this as a flat sheet, okay, so that we can study the 2 interactions. So what does this look like? So here's our experimental platform. This is a regular Petri dish, culture dish, and we have a device suspended in that dish, covered with a scaffold of whatever design that you choose to be looking at. And then these are our pigment epithelial cells growing on top. So this is with the apical chamber. This is the basolateral chamber or choroidal chamber. The cells know how to polarize. They know which way is up, because the polyester is coated with proteins that are found in Brooks membrane. So the cells settle down. They say, oh, this is bottom. I know what to put on the top of my cell and the bottom. So the cells polarize themselves. Now, what can we do with this setup is we can put electrodes on either side, and we can measure the movement of current across this monolayer, or the resistance to current. and measure it with an ohm meter. So high resistance means low permeability. We want a low permeability cell because these are to form a tissue barrier. Okay. We could also put a tracer here and see if it moves to the other side, or do the opposite experiment. These cells are filled with pumps and channels and transporters that are designed to push nutrients to the photoreceptor cells and to take wastes and remove them away. So we can examine this tissue to see if those kinds of proteins are there, and are they in the right place? Are they in the apical membrane or the basolateral membrane, as they should be? So we have different kinds of scaffolds that people have explored. So now we're looking down on the scaffold. These polyester membranes that I've been talking about have been a workhorse in the field of epithelial biology. And the way they're made is you poke holes through them using a radioactive source. So this is an etching technique. And you can see the pores have a defined size and they're randomly distributed. This hexagon in the last earlier slide of the RPE, looking down on it, you saw this polygonal network with the stain that labeled the cell borders. Maybe we go back to. So these aren't perfect hexagons. In life, they are. But in our artificial culture system, we have more irregular polygons, but it's the same idea. Okay. And so I put this lattice on top to give you an idea of how many pores there are per cell. Someone used a polycaprolactone. It's found in suture material that surgeons use for internal sutures that need to dissolve. PET polyester won't dissolve, but these will, and that's a desirable thing for a transport tissue. What we want is the scaffold to disappear and be replaced with an authentic Brooks membrane. We want that Brooks membrane to be synthesized in the human or the animal model. They engineered a uniform distribution of pores that are slightly larger. Another technique is called electrospinning. PLGA, doesn't matter what it is biodegradable. And this is an enlargement. So this dot is the same size as one of these pores. Okay, so it's just a higher magnification. So this would fill the cell maybe about like this. And you can see these electrospinning is a kind of weaving technique. You make this very thin fiber of your scaffold material, and you essentially weave it across a supporting structure, and you create this meshwork. So we have all these pores, and they're uniformly distributed, and it turns out we get the best cell morphology if we grow it on this kind of filter. A lot of the biochemistry and cell biology of the RP is reproduced on these other filters, but this one does the best job of that. So how about the retina side of this? So the technology for making a monolayer of any kind of epithelial cell is decades old. Okay, people know how to do that. We don't know how to do the retina as a flat sheet. So what we did is we made a block of tissue composed of these compounds that are found normally in the neural retina. They're the proteins, the extracellular matrix is, think of it like soup. It's the water in your stew. Okay, so the cells are the stew and the green beans and the peas and whatever else you put there, and the surrounding fluid, we would call that the extracellular matrix. And so we're creating an extracellular matrix similar to the retina. And we make this as a block. And so here's a cube of this material in the scanning electron microscope. I've put dashes to emphasize the edges of the block. In this image, we're looking straight on a block face. And these pores are enough to accommodate 10 cells across, more or less. Okay. So we're going to take our retinal progenitor cells um, before they begin to form all these different layers, and we're going to dissociate those cells and seed them on this. What we'll do is cut off a 60 micron slice off of this. And so what it ends up looking like is this. So here is my slice of tissue, of scaffold seen on end. The orange dots are the retinal progenitor cells. The cells that are pushed, they're going to become retinal cells, but they're not retinal cells yet. Okay, so the retinal progenitor cells. And we're going to put this mess on the culture of retinal pigment epithelium. And we're going to ask the question, does the pigment epithelium help these cells mature? As these cells mature, they'll feed back signals and help the RPE mature. And so you'll have this crosstalk, kind of like bootstrapping a computer program. That occurs with the neural retina between these cell layers. We know that goes on. So our postulate is that this kind of interaction will go on simultaneously with this kind of interaction, and we'll get a multi-layered sheet. So now we might not have a transplantable tissue, but we will have recreated an experimental platform so we could try to understand the cell biology that underlies the tissue that we ultimately want to transplant. So here's the experimental setup again. I've created this pseudo subretinal space. The notion is that anything that's secreted by the RPE has to concentrate in this space. It can't get diffused in the bulk of the media chamber. And the same thing for things secreted by the retina. Okay. So here's what our retina looks like, our pseudo retina. And we can see this is labeling cells that will become photoreceptors. These are stain labeling cells that should be in the layer adjacent to the photoreceptor cells. So we're starting to make some progress. The problem is, compared to a retinal spheroid, we mimic what happens in a retinal spheroid for up to about 3 months, but then from 3 months on, we're not making progress anymore. And so we could talk a little bit about what we've learned about that, which I think is critical, because I think we're going too fast into transplanting tissues into patients, not really understanding what's going on with the cells. If we do this in a pig or a rat, at the end of the experiment, we can sacrifice the animal and we can do any number of kinds of analyses to figure out what actually went on. If we're using purely non-invasive techniques, because most people don't like their eyeball taken out and sliced and that kind of stuff. There is a lot you can learn looking through the lens, but not enough. Okay, so developing experimental platforms, I think, is going to be very important. So if you look at this 3-month time point when things are going well, and we do an experiment called quantitative RT-PCR. So what's that? The polymerase chain reaction is a way of measuring mRNA in a cell. The mRNA is what is going to be transcribed into protein. So we're looking at gene expression. And so we can grow cells as a monoculture, either RPE monoculture or retinal progenitor cell monoculture, and we can do the co-culture. And so we're comparing expression to a housekeeping gene. Housekeeping genes are expressed by all cells and in high quantity. And so every other gene we're looking at is expressed in lower amount. So this is a base 2 log scale. We can talk about why we use base 2 if you care later. So this is 1, same level of expression as the housekeeping gene. And if expression is constant between the monolayer and the co-culture, we should fall along this 45-degree line. And we could see for the RPE, a number of these important RPE proteins are expressed more than fourfold more in the co-culture. So it seems that the developing immature photoreceptor neurosensory retina layer is benefiting, promoting the maturation of the pigment epithelium. If we look at the other way, nothing seems to be going on here. We can't make any sense of why some genes are downregulated and other genes are upregulated. If we look at proteins themselves, the proteins that actually change are opsins. Opsins are those proteins that capture light. So rhodopsin captures all wavelengths. Color cone opsins, short length, long wave, and intermediate length light. So we have those four basic types of opsins. And those seem to be affected by culture with the pigment epithelium. If we look, oops, come back here. Look at our electrical resistance. And so this label is wrong. We're looking at the resistance to a current across the RPE monolayer. If we look at the resistance of the retinal progenitor cells, it's virtually nothing. It doesn't offer any resistance. Our RPE offers a bit of resistance, but if we do the co-culture, resistance goes up. So another indication of functionality, the nascent progenitors of the neurosensory retina are helping the RPE mature. And we did this with 3 different cell lines. So that's important, because when you're looking at stem cells, you have to ask yourself, well, is this an interesting finding, but it's peculiar to this one particular preparation? So if we isolate stem cells from 3 completely different sources, and we see the same phenomenon, then we have some confidence that it's real. So what we talked about, the macula is just a small region of the retina that lies along the main visual axis for visual acuity. Phase 1 clinical trials are demonstrating safety. Phase 2 clinical trials, perhaps soon, will recruit patients who are more likely to respond to the transplant. And the challenges are we have this complex outer retina. It's this computer chip wired in ways that we don't entirely understand. And we've got to get it to reorganize. And will that just happen spontaneously? And what are the things to put in the eye that will promote that reorganization? And my thought, the thought of my collaborators, is that we need to do more work in culture with these culture models that I described to you, with animal models that I didn't say very much about. But we need more work there before we can move to phase 2 clinical trials, so we can understand why things are working and why they're not working when we put them in a patient. So with that, I'll stop and take questions.
So Larry, I'm familiar with drug development where any company or any person or drug registers the clinical trial. So the process is to register the clinical trial on a database called clinicaltrials.gov.
Yeah.
And in order for the trial to be considered for approval by FDA.
Mhm.
Does this go through that same process? Would people doing phase 1 or proposing a phase 2 trial put that information that they're doing the trial so that a potential volunteer could get it? Will that go on clinicaltrials.gov?
Yes, absolutely.
Okay.
And if you go there, they'll find, I don't know, hundreds of trials going on.
So one of the questions people have is, what's going on? Can I get into a trial?
You can.
That's how they would do it.
That's how they would do it.
There's a website out there that you can access, clinicaltrials.gov, that will tell you all the trials that are going, or at least most of the trials that are going on in this field.
And Add to that, you can go to your ophthalmologist and ask if he knows of a trial. They might be participating in a trial and know someone who's participating in a trial. To do it, just make sure you're going to a major medical center. There was a horrible story of a clinical trial that was approved and then withdrawn. But a small clinic decided to do the procedure anyway. And it was done by nurse practitioners, and it was done in it was not done in a hospital. And they had complications. Fortunately, they were near Bascom Palmer in Miami, one of the foremost eye centers. But unfortunately, the time it took to get there, the patients were already blind. And they did both eyes at the same time. So, it's a buyer beware. Kind of make sure you're working with quality people.
Are the stem cells patient-specific? And if not, is there any problem with a transplant rejection type of phenomenon?
Yeah, so immunosuppression, of course, is a big deal, and so there are a couple of approaches to that. They tend to be stem cell lines that are well established. We know how they're going to behave. Although you think there's stem cell lines they can give rise to any kind of tissue, some cell lines are better at certain things than other stem cell lines. Okay, so that's one issue. So you can take the stem cells from the patient in principle, and that's a lengthy process that may or may not work. But what people are trying to do is make stem cell banks, hundreds stem cell lines that are characterized according to their immunogenicity. So you could say, oh, this patient needs that particular stem cell line, so we're going to make our tissue from that to try and minimize the need for immunosuppression. But yeah, it's an important problem.
Is there, from a science fiction point of view,
Yes.
is there any work going on a biomechanical method of seeing when all is lost, you haven't got your sight at all?
Okay, so actually there are patients walking around with computer chips in their eye. There is a limit to the density of photocells that you could put on a chip. Okay, so the resolution is not great. Okay, so a notion is if you put the chip in the subretinal space, then maybe you could take advantage of the neurocircuitry to improve the resolution, because there's a physical limit to the resolution of a chip. It turns out that nice in principle, but it doesn't really work that way, because the electrical field developed by the chip spans all layers of the retina, no matter where you put it. So patients are not reading books; they're not reading the daily, but it's qualitatively it's a big improvement in their lives. Okay. So those are still not ready for prime time, but they're kind of wild things. You've got little mirrors in front of your eye that are amplifying the signal so that it's strong enough to stimulate the chip. But yeah, there's progress there.
How is the wet form of macular degeneration different from the dry form?
Okay, that's a great question. Wet form versus dry form. So the wet form can originate with the RPE, or very often it originates with the choriocapillaris. So I showed you that cartoon of the RPE with the Brooks membrane and the choroid underneath it. So what happens in wet macular AMD is those choroid vessels start growing aberrantly and poke their way through the RPE and into the subretinal space. Okay, so now the retina is detaching, and that subretinal space has become a real space containing leaky blood vessels. Okay, which is so now you don't have a blood-brain barrier anymore, number one, and you don't have this intimate contact of photoreceptors in RPE microvilli. So vascular endothelial growth factor was named because it's a protein that promotes the growth of endothelial cells. So the idea is to counter that with an anti-VEGF, vascular endothelial growth factor. So anti-VEGF can be injected into the vitreous as small so it can diffuse across the retina, get to the subretinal space and inhibit, and if it's caught early enough, help regress those aberrant capillaries. So that's the only form that we have a therapy that does any good. There are a lot of people walking around getting their monthly or 3 or 4 times a year injections and are doing pretty good. The dry form, we really have nothing. There's a vitamin therapy. If you're starting to show signs that you could do this vitamin therapy, that might help a little bit, but we're really at a loss for the dry form in terms of therapies. I think you might have, I am, but where it says Mike, there's a little button to push. It's like a power on and off button. Yeah.
Yeah, you're talking about ILEA, and
Yeah.
what about ARIDS2? That's what you're also discussing?
Yeah, so Alea is one of these anti-VEGF preparations. There are a couple of floating around. And the ARIS-2 was the study that developed this vitamin antioxidant therapy that has marginal success with helping dry
So,
AMD.
I'm an optometrist. I've been dealing with this for 50 years, and for 50 years, there hasn't been much. How many years are we away from the stem cell?
How many years are we away from the stem cell? I'm reminded about a chairman of surgery that in my department talking about xenotransplantation, and said xenotransplantation is 5 years away and always will be. I hope it's not that bad. I'm not sure that the therapies or the approaches that I described here are going to be the magic bullet. I think they're going to be better as experimental platforms to figure things out. But injecting a cell suspension is an easier thing and might actually work better. So there are problems with that, but people, so it kind of fell out of favor, but people figured out solutions to that problem. And so suspensions are coming back into therapy. So we've got competing approaches that people are working on. We're getting somewhere. We're getting somewhere. So, and we haven't stopped yet. So I'll take out my crystal ball after I have a few drinks later tonight. And yeah.
Good morning.
I'm sorry. Yeah.
I have wet in my left eye and dry in the right.
I see.
I receive injections approximately every 8 weeks in the left eye. I also have developed a cataract on that eye. Is there a possibility that if I had that cataract removed, that if I had the cataract removed, that it could injure?
Okay, so the question that faded away. First, I'm so sorry to hear of your disease and complications and so forth. So in principle, so the idea was we have macular degeneration, we have a therapy that's working. We developed a cataract, and is moving the cataract going to affect the macula? And I meant to ask this at the beginning. Are there any ophthalmologists in the group? Oh, thank God. Uh, okay. Um, that's certainly a question for a medical doctor. I like to tell people that, my kids used to say, oh, my daddy's a doctor, but not the kind that can help you. So, but in principle, those are opposite ends of the eye. You should be able to replace the cataract. And whatever's going on with your retina will still be going on. If you're, well, I could talk to you more later. So it's the eye where you're getting injections, the wet eye. Yeah, I would say likely not. Ask a real doctor. I see. Yeah. Yeah. So I would get another opinion, and I would go to some place like Johns Hopkins, the Wilmer Eye Institute, or first name in UCLA. What is the actually? No, with John Hopkins is in Baltimore. But when I think of the top ones, I think about Johns Hopkins, Baskin Palmer, and Jules Stein in UCLA. So you see what they say about it. Yeah.
Instead of, let's say, monthly injections, is there any progress in coming up with some eye drops instead of have to go to the ophthalmologist for injection?
Wet form, I'm not aware of that. Fortunately, the eye injections, they sound, oh my God, but they're really pretty painless. Yeah.
Further question, what initiates macular degeneration? Do we understand them?
No. So what initiates macular degeneration? It is a multifactorial disease. There's a genetic component to it in some cases. Smoking, diet, other comorbidities, cardiovascular disease. There are 100 things that play into it. So understanding the pathophysiology is one reason for developing culture models, and another reason for doing more work in animal models to figure that out. People are not an experimental model at this level anyway. We have a question over here. Right here. Well, it helps for the recording.
Okay, put them out. Yeah, actually, you probably could hear me better without this. No, that's amazing. What I find is that for dry MAC, there's really a genetic component, and what you find is that you can identify that you have the gene for dry MAC, if you look at families, for example, in my family, you'll find out that several people have developed macular degeneration because they essentially have the gene. Now, there may be other effects that would cause this dry MAC, but definitely, I have dry MAC, and my son, by the way, turns out to have dry MAC, or has a dry MAC gene. So he's kind of working kind of heavily with me to find out what the solutions may be, because he suspects that he's going to end up with the same problem. So-
Might well be. I think there are, the last time I looked at it, something like 34 genes that are associated with macular degeneration. But I'm not sure I would characterize any of them as the macular degeneration gene. And it's what are the co-factors, the co-risk factors that go with it? That's very active area of research. But families like yours, my heart goes out to you, but families like yours are so very important, where we can trace a lineage, how our genes passed, and who gets it, and who has the gene that gets the disease, and who has the gene that doesn't, and that's very valuable for researchers. I hope you're connected in with people that do genetic studies in macular degeneration.
On that same line, if it's identified already in a child of age 1 or 2, and there is that factor that an older sibling has it, is there any danger in just waiting to let the child continue to mature before you can find somebody who could offer some alternatives? Because like in the Buffalo area, there wasn't anybody to find, and you know, how far do you have to search, and can you wait? The child is now 3. So is time a bad factor?
So, so the questions become: are you talking about a juvenile form, or yeah? So it's a so a disease like Stargardt's disease is one where there is a age-related like thing. There's a single gene, and we know what that gene is, and people work on gene therapy approaches for that. I don't know what gene you're mentioning, but juvenile onset is a macular-like disease that occurs in the 20s or 30s of individuals, so that's much simpler than age-related macular degeneration, because we can identify this is the gene. You have the gene. Maybe it interacts with other things as well, but there's a strong tie. It's a single gene disease, which means a single gene therapy. You know what gene to create, to inject, to correct the problem. Okay, so there's a different therapy route that people are working on for that kind of simple thing. I put in quotes simple thing. Retinitis pigmentosa is another collection of hundreds of diseases, but they all involve a mutation of a specific gene. So if you know this specific gene, you have a target to go after. It's kind of like the COVID-19 virus. Once they had the sequence, they had a target to go after, and they developed a vaccine very quickly. Gene therapy is a little more complicated, is different than that. I don't know the status of clinical trials for that kind of gene therapy.
Thank you, Larry. I have, please.
Thank you.